Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and today I'm going to talk about
nerve pathway for good and bad feelings identified. Scientists in Japan have identified a nerve pathway involved in the processing of rewarding and distressing stimuli and situations in mice. The new pathway, originating in a bundle of brainstem nerve fibers called the median raphe nucleus, acts in opposition to a previously identified reward or aversion pathway that originates in the nearby dorsal raphe nucleus. Previous studies had already revealed that activating serotonin-producing nerve fibers from the dorsal raphe nucleus in the brainstem of mice leads to the pleasurable feeling associated with reward. However, selective serotonin reuptake inhibitors or SSRIs, antidepressant drugs that increase serotonin levels in the brain, fail to exert clear feelings of reward and to treat the loss of ability to feel pleasure associated with depression. This suggests that there are other serotonin-producing nerve pathways in the brain associated with the feelings of reward and aversion. To further study the reward and aversion nerve pathways of the brain, researchers in Japan focused their attention on the median raphe nucleus. This region has not received as much research attention as its brainstem neighbor, the dorsal raphe nucleus, even though it is also a source of serotonergic nerve fibers. The scientists conducted a wide variety of tests to measure activity of serotonin neurons in mice in response to stimulating and inhibiting the median raphe by using fluorescent proteins that detect entry of calcium ions, a proxy of neuronal activation in a cell type specific manner. They found that, for example, pinching a mouse's tail, an unpleasant stimulus, increased calcium-dependent fluorescence in the serotonin neurons in the median raphe. Giving mice a treat such as sugar, on the other hand, reduced median raphe serotonin fluorescence. Also, directly stimulating or inhibiting the median raphe nucleus using a genetic technique involving light led to aversive or reward-seeking behaviors such as avoiding or wanting to stay in a chamber depending on the type of stimulus applied. The team also conducted tests to discover where the switched on serotonergic nerve fibers of the median raphe were sending signals to and found an important connection with the brainstem's interpeduncular nucleus. They also identified serotonin receptors within this nucleus that were involved in the aversive properties associated with median raphe serotonergic activity.
That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.